of us have already taken. But if you haven't seen any of this content before, that's absolutely fine. We're going to cover this as if this was essentially one of your main core classroom sessions. So let's start out with complex diagrams. For example, if we saw a discrete question like this about titration of aniline hydroxide, of course there's a nice transition from John's session a second ago, thanks John, we might immediately have some hint that, okay, we're doing the titration curves, we want half equivalences or whole equivalences, but it's a relatively complex diagram because we have certainly some amount of value being added for equivalence. Then we have to read the relationship here off of the graph, find how it relates to the PI, but we don't get to draw on the graph like I'm getting to do right here. Instead, we have to approximate, we have to do a ballpark answer. So hold on. Our x-axis is equivalence, our y-axis is pH, and we should pay attention to a couple points. Anytime we have values on our x and y axis, this is true for the vast majority of questions we'd ever see on the MCAT, we want to pay attention to what units we have and how the axis has actually been labeled. Additionally, if it's a really complicated diagram, it's an apparatus, or it's a long process, or it's some sort of flow diagram, we might be able to swing in, grab the point on the question by thinking about it conceptually without necessarily spending a long time analyzing the diagram. So as soon as I see a question like this, it's somewhat complex, we could do one of those two pathways. Now in this particular question, it wants to know, okay, what would be the net charge on most aniline? And the answer choices are all negatives or positives or a perfect zero. So glancing at this diagram, clearly we can see that there's a first pKa, there's a second pKa, so there are two main points where we have a plateau, a buffer region, and there's one pi value where we have a steepest point on this curve. Luckily for us, the value they're asking about, this 1.2 equivalence, looks like it's somewhere right there at about that steepest point in the curve. To be clear though, on the MCAT we can't touch the screen, we can't draw on the screen, we can't copy this down necessarily onto our scratch sheet and get a really good feel for it, so we have to approximate based off of essentially a rough guess. So help me out down here in chat. Where about does it look like this line is falling within? Within the pK2, within the PI, what is it closest to? It looks like it's closer to the PI than to the pK2. Absolutely. Alexis, yes. Andrew, yes. And that's the point. On the MCAT, we don't actually have fine gradations between these values. We can't know for sure where it's falling, but we have a pretty good approximation. It's still on a steep portion of the curve. It's definitely not hit that buffer zone plateau. So if it's at the PI, well, PI is when we have an equal amount of positive and negative charges, which is this electrically neutral molecule, which means that we're really happy for our aniline. Well, then it's got to be answer choice B. It must be the case that we are right there at that zero, at that perfect point. Remind me, what would we call it if we had an amino acid at its perfect PI value? It is isoelectric point. Absolutely, you're right, Alexis. That's what PI means, isoelectric point. And there's a special name for the molecule when it's equally broken down, perfectly neutral, with a net charge of 0. Yeah, the Zwitter ion. Nicely done, Alexis. Absolutely fast responses. So we have, of course, our negative charge on one terminal and our positive charge on the other terminal, but overall a net 0. Nice. Now, aniline, of course, is a molecule that we should be relatively familiar with. And if we're talking about equivalence of hydroxide added, we should also be aware of whether alanine has multiple protons it could even have released. So help me out. Is alanine one of our overtly acidic amino acids? Does it have multiple acid substituent groups? No way. Alanine does not. Bonus question, what is the side chain on alanine? It's one of our shortest amino acids. It is one that hopefully we would have memorized by test day, even if you don't have it already. Nicely done, Alexis, Andrew, fast responses in here. And again, absolutely, we have a methyl group as our side chain. Methyl group is not going to have extra acidic protons. So again, if there is any sort of quick relationship to alanine, if we already have it memorized, we can use that to eliminate answers. Now for 18, let me talk about what I just mentioned a second ago. Reading the graph efficiently, to try and get the point out of a complex diagram. Or the second thing I mentioned was potentially being able to use some critical reasoning to shortcut having to do the complex diagram analysis and instead figure out generally what's being asked. Well, so for number 18, we're generally being asked about hydrophilicity and hydrophobicity and about the pathway. Gosh, there's all these spiky things coming through here. We would potentially, want to figure this out without having to read every one of those peaks. This looks sort of like a discrete question again. It's got another curve. It's got a lot of values for amino acid residues and hydropathy index, but looking at the graph, it actually mentions positive values and negative values in the question stem. So if we're looking for membrane spanning regions, then we're looking for that hydrophobic region set, and everything positive, they tell us, is hydrophobic. So we have two regions that are above the axis. Done. It's choice B. But this is really a conceptual question. We would have to remember that the membrane region should be hydrophobic. We don't want water inside that membrane region. So there must be these two intermembrane spanning regions, these two chunks that are above the zero. Now, they could have asked us about a lot of things within this question stem. For example, they could have asked us how many hydrophilic regions do we see? Bonus question, just to make sure we understand the critical thinking behind this. How many regions would we expect to want to bond out to water? 3. There are 3 groups of regions, absolutely, Christopher, Andrew, everybody, nicely done, that are right there between each of those hydrophobic regions. Now, about the trick we used here, again, to really try and lock in what's being asked for, we want to think, is this a conceptual problem where I have to understand groups of information? Is this a specific problem where I have to actually read a particular value and I have to translate it? What is being asked for with a complex diagram? And that becomes very important when we see multiple diagrams or we see relationships between diagrams, for example, in this one that you had later on in the test. Protein synthesis by the egg types in the experiment. Now, if you don't remember this one from the test, or if you haven't had a chance to take the test, then this relates back to a figure from the passage. This is the 4th passage in that set. And the passage told us that protein synthesis, there are 3 different types of sea urchin eggs that were being experimented on. Whole eggs, half eggs that had a nucleus, and half eggs without a nucleus. So we'd have to try and figure out, okay, if we're dealing with protein synthesis, what are we really talking about? What is actively translated when we're making a protein? This is a conceptual problem, content-based problem. I pose it to you. What is being translated when we're making a protein? Yeah, mRNA. Perfect. Remember, transcription, translation, all of the process of DNA replication, we should have a pretty good feel for, because the passage might briefly run through it. Might talk about meiosis or mitosis processes without ever saying meiosis or mitosis, and we have to recognize that. Same thing with transcription or translation. We have to be able to key in. If we're talking about translating this, then we're talking about mRNA becoming a protein. Now, if we're talking about mRNA becoming a protein, then the question is really asking about the protein synthesis that would be possible in each of those 3 types: a whole egg, a half egg with a nucleus, and a half egg without a nucleus. And we want to think, well, in what case would we have a finite amount of mRNA so we would see some sort of dropping off? Now, the reason that we pose this question is because in the actual passage, you only got a small piece of information. You just got this stuff down here at the bottom. We're talking about activation, and it seems like the curve is going up. But this new passage information that is now in all of these answer choices clearly shows a plateau or a drop in the vast majority of the answers. So that's why we would pose this second question. Which of these potentially would drop down, since clearly all of them have some sort of plateau or drop-off? The tangent question to that would also be, which ones shouldn't have a fixed amount? So help me out down here in chat. Which one would probably have a problem? The one with no nucleus? No nucleus. That sounds like there'd be a, gosh, pretty hard time with mRNA production in order to make those proteins. The half egg without a nucleus doesn't have DNA, won't be able to supply the necessary new mRNA needed for translation. Without any influx of mRNA that the DNA transcription supplies, translation would run out once the original mRNA is consumed. Awesome. Absolutely right, Quinley. So looking at the answer set, we'd expect whichever one matches up with half egg without nucleus to at least dip down. Well, looking through the answer set, again, a little bit hard to read off of this diagram, perhaps, depending on the resolution of your screen, but I expect that little teeny tiny dotted line to be one that dips down. Well, that happens in A, absolutely, Neva, Christopher, Andrew. It doesn't happen in B, it doesn't happen in D, so we can get rid of those too, but it also does seem to happen in C. So we can't immediately lock on to A yet. Remember, every time you do a step of work, make sure that we at least give ourselves some credit for it. At least cross out some answers, but don't go too far unless we actually know it. So half egg without a nucleus, we determined it has to be a downward dip. The follow-up is, okay, no nucleus makes no DNA, makes no mRNA. Is there any other way we could run out of mRNA? Could, as we show here, the whole egg run out of mRNA and suddenly dip down like we see here in answer choice C? As far as we know, in a normal whole egg, no. I had to add that piece because the MCAT assumes that content. Until you know that something is going wrong, you always assume that things go right. Remind me, what hint might they give you in the passage to tell you that this experiment might not have everything going right? What hint do they often give us? Yeah, exactly, Quinley. Not us as students, obviously, don't take offense, but the students, quote-unquote, the AAMC finds somewhere, apparently make a lot of mistakes and make careless errors, so if we had been told, hey, a student is running a protein synthesis experiment, then yes, potentially we might get tempted by thinking, hey, maybe the whole egg has a problem, maybe the half egg with nucleus gets a problem, maybe there was some contamination, maybe something happened. Again, we don't necessarily know going into this particular subset of questions, but if we had seen a student, maybe we would have thought an error could come up in a later question. But just to be clear, unless the MCAT tells you there's an error, we assume there isn't one. We assume the whole egg is perfect. There is no possible way that a whole egg with a nucleus could have a downward dip like C does. So part of C was right, but the other part of C we have to throw away. Now, going one step further in this concept of complex diagrams, remember, anytime we see a brand new diagram or a graph or a figure, look for units, look for labels, look for relationships. For example, in this one, this is the first graph problem we've done so far that doesn't have any values written in here. It's completely conceptual. We sort of transition from the first problem, dealing with actually looking at the number and following it on the graph. The second question is dealing with regions, to this third question, totally conceptual. Which one would have some change of dipping down versus increasing compared to the other one? With that, let's transition into how the passage questions, besides just these discrete ones, can be quickly determined. Because often, a passage might have, like these past three questions, where they're essentially pseudo-discrete. We didn't even really need to know a lot about sea urchin eggs in order to make the connection to the fact that translation had better have mRNA becoming a protein, and if mRNA becomes a protein, then that needed a nucleus at some point originally. So we could have gotten the answer to that one, actually, if all we knew was the different eggs, nucleus or non-nucleus eggs. We'll see a lot of these throughout the MCAT, specifically because they might give you a passage that gives you a lot of information about membrane movements and umami as a sense organ on the tongue and a sensation on the tongue. So maybe we would see a passage about receptors on the tongue and about umami versus bitterness versus sweetness, and they get a question like this, which is about glutamate, right? Which glutamate receptors relate to the umami sense. It is one of our amino acid general aid receptors on the tongue. So this could come up in that passage, but it's clearly a discrete question. It's basically asking, how do ions move through an ion channel? If there's an ion channel and we're putting stuff through it, we have to know what that means. Basic Bio 101 material here. Help me out down here in chat. How do ions move through an ion channel? Yeah, they go down their concentration gradient, ideally Quinley, absolutely right. Now, what Christopher put in here, I'm going to talk about in a second. And I also want to ask a question. Was anybody significantly tempted by C or D, even if you didn't choose them? You were significantly tempted by those two answer choices. I ask because usually we have a large number of students who are. And if you weren't, awesome. Give yourself a pat on the back. You've got this down. But yeah, I mean, we'll throw some honest yeses in here, Christopher. Great. Let's talk about it, because this is a sort of learning experience everybody can benefit from, even if you were already feeling pretty confident on this problem. For question 13, they're asking about ion channels. Well, an ion channel specifically makes us think, okay, this is something particularly dealing with diffusion where the thing can't go through the walls or the membrane easily. If it was, then we would have passive diffusion, aka simple diffusion. So to Christopher's question a second ago, or maybe general question, passive always means diffusion that can go through the membrane or passively go down its concentration gradient without needing anything else. Just go through the membrane. It's small, it's nonpolar, it can slip through. Now, there are some things that are small and very polar that can slip through also. In the vast majority of every membrane question we would ever see on the MCAT, and those small things are water. As far as we are concerned, any membrane that you ever get in the MCAT will always have aquaporins. That's nice for us because we never have to ask ourselves that question. Water should always be able to get through every membrane. So osmosis, I mean, it is sort of a channel for something really polar, but an aquaporin is its own specialized channel subset, and as far as we're concerned, the MCAT doesn't ask about osmosis as if it was an ion channel. Osmosis is more this special thing that water can do that nothing else can do. So we're fine with it. It's one of the exceptions. We don't talk about it. Between B, C, and D, though, here's the thing. Every one of those could happen through an ion channel. That's maybe why a lot of people get tempted by these three answers. But if we're thinking about an ion channel is allowing something to go through the membrane that wouldn't otherwise be able to. So by default, it is facilitating the ability of that ion to diffuse through. So unless we know anything else, that is by default the most general answer. It is facilitated diffusion. Simple as that. Now that's kind of specific to the MCAT, asking you a question where multiple answer choices, if you knew some new specific information, could be right, but only one answer has to be right. So facilitated diffusion must be the correct answer. Now, what if I had told you moving ions through this channel utilizing ATP? Yeah, we'll get to that in a second, Peter. You're right. Utilizing ATP. That is specifically, you got it, Andrew, primary active transport. This is something you should be familiar with. Primary active transport hydrolyzes ATP, and it uses that energy in order to push the concentration gradient back up against where it wants to go. So, for example, we might have been burning a lot of glucose in our muscle cells, and we're running a marathon, and then we have sugar, of course, going down its concentration gradient, feeding into the muscle cell areas, and all of a sudden we start to get dizzy and nauseous. Uh-oh, ran out of sugar somewhere else. We're going to have to use excess energy to pump that sugar back up against its concentration gradient to hopefully keep us conscious. That's primary active transport, but we have to spend energy to do it. Wouldn't it be better if we had just hydrated with fancy electrolyte drinks? Yeah, This is why we hydrate with fancy electrolyte drinks. Because what if I had asked in question 13, moving, in this case, glutamate against its concentration gradient by allowing a different solute to go down its concentration gradient? Yeah, that is answer choice D, secondary active transport. There are two different things we could be talking about. We could talk about co-transport, where they both go across a membrane channel in the same direction. This is co-transport, or aka symport, co-transport or symport. And the other thing that could happen, symport. The other thing that could happen is going in the opposite directions. And this is antiport. Anti for going opposite directions, sim for going the same direction. So symport, antiport is a secondary active transport. Classic symport and antiport mechanisms would be, like I mentioned, salts and sugars. You can have sodium going down its concentration gradient and then have glucose hitch a ride there as well. Or you could have a sodium-potassium pump in neurons. Exactly, Christopher. That is an antiport mechanism. One goes out and the other one comes back. That would be secondary active transport. Now again, for it to truly be secondary active transport, ideally, we would have to have one go down its concentration gradient in order to slingshot the other one back up. That's important. So in neurons, for example, in one direction, we have the channels opening, we have a flood of the ions in the particular direction where they're going down their concentration gradient and they're happy. And in the other direction, by utilizing the pump, we actually have to use ATP in order to force them back into that resting potential of the neuron. So just being aware that if they talk about any sort of energy, that overtly means we're dealing with primary, because we have to use energy to make it happen. Secondary active transport avoids using ATP by slingshotting. Right, exactly, Christopher. Yep. And that's that fine difference. Again, that's why I wanted to cover this with you, because a lot of people get this question a little bit confused by overthinking or by trying to think about the possibly true as opposed to just what's been overtly given in front of us for the question. Now, another nice transition, actually, from that discussion of maybe running a marathon and glucose has to go against its concentration gradient, is this fancy thing where we make MSG from sugar molasses. Now, the passage actually gave us a little information from this, so it'd be relatively difficult for us to answer this maybe without the passage, but let's see how well we could get to the answer. Bacteria is producing something from sugar. What do you think, if we're dealing with bacteria, we are in? An aerobic or an anaerobic environment? What do you think, if we're dealing with bacteria? Sounds like in the general case, we are often in an anaerobic environment. Sure, absolutely, Quinley, Andrew, and if we're loving on some molasses, then what sort of a process are we probably dealing with? Probably dealing with sugar breakdown? Sounds like glycolysis? Absolutely, Quinley. So looking at the answer set, even without the passage, gosh, this looks like a pseudo-discrete. Sure, we could have gone back to the passage where it says fermentation of corn, sugar cane, blah, 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 but it's got to be glycolysis, and we're talking bacteria. Boom, answer choice B, this is a pseudo-discrete. We could go back to the passage, talk about fermentation, blah, 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 but there's a barrage of new information from that passage. I mean, a good outline might allow you to quickly recognize the detail presented, go back to that nugget of information, and answer the pseudo-discrete. But additionally, the whole point is figuring out, are there shortcuts we can take without having to go back and reread a whole bunch of text? Now, sometimes we would need to go back and research the text. For question 32, for example, we've got tumorigenic cells. Maybe the passage, from this case actually was passage number 7, is pretty dense. It's got a lot of information about cancers and about information about tumors in general. There's a bunch of dense information that we'd have to slog through to get to this question. However, tumorigenic. Sounds like kind of the root tumor is right there at the beginning of the word. So a tumor, remind me, tumors, what general class does that fall into? A tumor, aka some sort of cancer. Yeah. Now, a cancer on the MCAT is sort of a garbage heap of a whole bunch of things that are interrelated. But in general, for our purposes, cancer essentially means a few specific things are happening. Help me out down here in chat without even looking at the 1, 2, and 3 Roman numerals. What sort of things would we associate with cancer? There are only a couple, just to be clear. We can definitely associate with a cancer. Increased cell division, you got it, Christopher, growing really fast, exactly, Quinley. Those are the two main ones. Really growing fast, rapid cell division. Now, that sort of processing, and the other one that I might want to see in here is abnormal. There's no stimulus that would directly be making that happen, as far as we can tell. There's nothing else in the body that would normally create this sort of rapid cell division or this really high growth. Because there are times in certain stress scenarios or in certain lifetimes that we might actually see rapid cell division or rapid growth, but this is without a stimulus for it. Something weird is happening. So now we can jump into the answer choices with that in the back of our mind. But just to be clear, the MCAT does not expect you to know the difference between different types of tumors, between different types of cancers, between the types of treatments that we can employ for them. That is well beyond the scope of this test. However, what if I told you that there was a cancerous tumor in the pancreas? What might you expect some of the symptoms to mimic if there's excess cell division or excess growth in some regions of the pancreas? This is a bonus question. It's not even related to this one on screen, but it's just trying to lock you in. Pancreas. It's definitely involved in digestion. It has all of our excellent enzymatic juices. It's got bicarb that gets released. It releases some of our mediating of satiety. So it could have some sort of indigestion. Sure. I like that one. What else does pancreas involve itself in, since we're on the topic of food? Definitely, Christopher. Mass effect. Too many juices, too much response because we have too much division. Definitely. I like that answer. Bicarbonate would be, again, neutralizing our acidic chyme from the stomach. Maybe we produce way too much bicarbonate, or maybe we have a blockage due to some other parts of the pancreas growing too fast, and we have either too acidic or too basic in the small intestine. That sounds like bad news bears. We don't want to have some sort of lesions forming or breaking down the walls of the small intestine. And then, as I was kind of hinting at, you got it, Peter, right? And your insulin response, right? Metabolism in general, uptake of sugar and breakdown. If we have the pancreas being affected by some sort of tumor, maybe we're affecting our insulin response. Maybe we have a diabetes-like symptom. Again, this is the sort of question the MCAT could still ask you that relates to fancy tumors or fancy cancers that we don't actually have to know, by instead having it affect an organ or having it affect a pathway that then we could get to the answer from. So I stand by my statement. You don't have to know specific stomach cancers. But if you had a cancer that affected a part of the stomach, you would want to know what does the stomach have for enzymes or metabolism or for any sort of excretions, what could be being affected. We don't have to go into multiple examples, but I wanted to give you that insight. Now, for question 32, they just want to know, well, what happens if we have a tumor? Okay, you said we'd expect to see a whole bunch of rapid cell division and a lot of growth. So would cell division and growth have mRNA being made? Roman numeral 1. Roman numeral 1, just to be clear, is in 3 of the answer choices. Our Roman numeral strategy is we would check each one. So, yeah, absolutely. Roman numeral 1 is good. How about would we have lots of ribosomes having assembly on them? Or being part of assembly? Absolutely. If we're making more cells, we better be making a whole lot more ribosome functionality. So 2 is good. Now, as soon as we know 1 and 2 are good, we would, of course, have gotten rid of answer choice C as soon as we knew that 1 was good, and now we would get rid of answer choice A because we know 2 is in. We'd have to check Roman numeral 3 in this case. Cell division, that was basically right there in our prediction. Yeah, absolutely, all 3, answer choice D, move on. but we take it piece by piece, step by step. What do we know should come up in the answer? Look through the answer choices, match, done. At this point, we kind of talked about looking at discrete questions, essentially. All of the questions that I just showed you were from passages, actually. But none of them really required me to go back and reread the passage. So as a reminder, on the MCAT, there are absolutely going to be discrete questions, but the MCAT is going to throw in a bunch of extra discrete questions, essentially, within the passage-based question sets, because they have to cover so much disparate content within their scope of standardized test. They're going to throw in a couple that don't really require the passage in the first place. So be on the lookout for those. When you hit a question, ask yourself, can I answer this now? Or do I have to go back and do my outline? Or have to go back and get a number or a word from the passage? If you have to, that's fine. The majority of passage-based questions require some reference to the passage, either for a number, for an experiment, for a molecule, for a detail, but there are going to still be faster questions like these ones that don't even require the passage. Now, speaking of the pathway for how the pancreas could impact some sort of digestion or some insulin or metabolism response, let's also talk about those complex pathways you can see on the MCAT. Generally for the MCAT, whenever we're talking about biology in terms of systems, we tend to get a lot more system-based pathway questions, where you might get a question about Krebs cycle in general, or a question about digestion in general, or the circulatory pathway of the heart. Keep in mind that the systems-based questions often tend to have things going wrong. This is just classic of the MCAT. They want you to have memorized the content, the complex pathway, the system of blood flow through the heart, but then they want to give you a bonus question about a problem. So suppose you're a physician, you're working in a clinic, and somebody comes in with a pulmonary embolism. Now, you don't have to know pulmonary embolisms for the MCAT, but what if I told you pulmonary embolism means we have a blockage, we cannot get in to the pulmonary system of the heart? Where are we going to see a buildup of excess pressure then? Okay, good. Quinley, yes. Right side of the heart. Christopher, yes. I like that. Could we get even more specific? Boom! Christopher's got it. Bonus points. Right ventricle. Absolutely. That's where we would see the pressure. Because we should think in sort of our overall view of the mind, okay, of the heart, excuse me, we've got this pathway where we're going through the different parts of the system. Okay, we've got the right, and then we're going through the pulmonary afterwards, and then we've got the lungs, and okay, and then the left side of the heart. All right, well, if we've got a blockage in pulmonary system, we can't get to pulmonary system, so we're in the right, well, first of all, atrium, and then we're in the right ventricle. Okay, and then we can't get into pulmonary, so we can't get into the pulmonary vein or the pulmonary arteries. All right, well, then we're backed up in the right ventricle. That's where pressure would increase. That's got to be our answer. And the MCAT loves to throw these process-based questions at you, where essentially you had to know the correct pathway of the heart to get that question right. But because I made it about a problem with the heart, it makes you do a little bit more thinking than you otherwise might have had to. So in this question, we're being asked, based off of the passage that you were given in the first place, about something that would be similar to the RAS activity. Now, in this question, there's a little bit more information. They're asking us about the RAS activity, what would be similar to it, but they're not actually asking us about the one from the passage. So if we have a cascade, or we have an activity that follows a cascade, what does that generally indicate as far as the overall system? What are we talking about when we have to have a cascade in order to see an effect at the end? One thing has to go to another. That is a secondary messenger system. Absolutely right, Quinley, for sure. Anytime we see cascade, it should immediately make us think that's got to be some sort of second messenger system, or it's at least a classic harbinger of second messenger systems. There are a couple other systems that might look like second messengers. For example, in this question, tell me what was the most tempting wrong answer? Maybe you didn't choose it, but the most tempting one that you were down to in question 34. Probably not A, it's an immediate response system. Probably not B, sodium-potassium pump is a functionality that does not require multiple messenger systems. Usually people get it down between C and D. But, just to be clear, D, any second messenger system should be our immediate prediction in our execute step, so we'd assume D. But Krebs cycle often is tempting. Remember, Krebs cycle, though, has a lot of different pieces, but we don't require that primary then secondary messenger system in order to actually have the process work. It's got a lot of moving pieces, but not ones that are required to signal in a second messenger process to the next. To be clear, this was one from passage 7, going through the biochem section, and we would have to glance through, in this case I think paragraph 2, to read about RAS activity, and a decent outline in the first place would have given us what RAS means. From the passage, we got that it was a cascade system, so we'd think second messenger system, and be able to get to answer choice D only with really a very vague understanding of the process from paragraph 2. But this one, obviously, is a passage-based question. We would need to have gone back, found that detail in order to really get this point. Same thing, again, with the next one that we're about to do. Let me show you the passage that this came from, just so we're all on the same page. This is passage 2, especially if you haven't seen this passage. Okay, this is about secondary messenger systems. This is about GMP, blah, blah, blah. Exactly right, the Viagra passage. Correct, Christopher. Now, as far as the PDE5 stuff, cGMP versus GMP, hopefully we would have jotted down something quick in our outline about the different pieces that are at play here, because as soon as I see a passage that has a whole bunch of letters in here, PDE5, cGMP versus GMP versus ED, there's a whole bunch of stuff to track. I don't want to get those confused. I want to know what is the difference between cGMP and GMP. What is the difference between PDE5, and is there another PDE I have to keep track of? What's happening? So anytime we see that sort of differentiation, immediately jot that down in your outline so we don't get confused. Now, as a nice bonus, we have tons of information here in our figures. To be clear, we don't have to really memorize a whole pathway because the pathway is in front of us, but we should notice some things that stand out as familiar. Oh, arginine, I've seen that somewhere before. Oh, here's my process with that cGMP going to the GMP. Apparently PDE5 is somehow helping that process happen, because otherwise we would have gone down through here to the other process. Okay, there's some stuff here I can come back to, but I'm not going to memorize the content from this pathway. I'm going to go back to this pathway if necessary. As a follow-up question, as soon as you see this figure showing you the structure of cinetafil, what do you immediately think we might get for questions? Even if we didn't from this particular passage, what would you think of that we might get for questions? Yeah, functional groups, because we see a couple classic functional groups. Absolutely, Quinley, Christopher. What else? Maybe a hint is there are some double bonds in here that are alternating with single bonds. Maybe about resonance structures. Sure, Christopher. Or about conjugation. What is the longest conjugation in here? How long do we have conjugation? Or what if we had a positive charge on this piece? Where could it resonate to on another atom? Absolutely. I clearly see we have some aromaticity in parts of this group. We have a long conjugated chain. We could definitely see questions about that. So remember, biochem absolutely has a good overlap with organic chemistry. So we certainly could see stuff about cycles, which we see in the bottom right of the molecule, aromatics, which we see in the middle of the molecule, functional groups. This is ripe with potential MCAT questions. Now let's jump in with a couple of them, though, that really showcase that concept of using either passage-based information or quick, discrete content. But for number 6, help me out. Do we know the pathway associated with this already, or do we have to go back to the passage? Go back to the passage. This is not one of our digestion, circulatory system, respiration systems that we should know going into the MCAT. Clearly, it was something from the passage, and it absolutely was there from figure 2. Here is that information pathway again. We'd have it, of course, on the left-hand side of our screen on test day. We absolutely can refer back to it. Now, it wants to know what best explains the lack of the sexual stimulation being necessary. Okay, so that means when they say best explains, only one of these is going to actually explain. The other three might be possible, could be tempting, might make sense in real life, but don't actually explain the pathway that we see. So tell me down here in chat, was there one answer choice that you immediately got rid of because you knew it's not even at all related to where Viagra is acting? What could we probably get rid of? Christopher said get rid of A. It's not expressed until the cavernous nerves are stimulated. Now again, we've got something about relaxes smooth muscle, corpus cavernosum, stuff like this happening down here at the bottom. But PDE5 is not expressed until that is... PDE5 is up here at the very beginning. That's in the wrong place in the pathway. So we would want it down here? No. What else could we possibly get rid of that is specifically not related to where we should be at for the activity of Viagra? Let's get rid of D. Thank you. Absolutely, Peter. NO? NO is way back up here in the pathway. Is Viagra acting up there? Is that where we see the sildenafil citrate? No. Not anywhere near that beginning part. So it's either B, Viagra does not produce cGMP, or PDE5 cannot be competitively inhibited under low levels. Now, I often recommend this to all of my students, but when you get to a question that's asking you about something that must be true, look for answer choices that are characteristically false statements. If we have competitive inhibition, can we overcome competitive inhibition? Yeah, it's the inhibition that we can get over. Absolutely. So C saying we can't overcome competitive inhibition? Now, certainly, increase in concentration is the way that we would overcome them. Exactly, Christopher. So C could have been tempting because you're thinking, well, it's low levels. Low levels means that, you know, it's going to be hard because we're supposed to have high levels of concentration to overcome. But we certainly can overcome competitive inhibition. This cannot be competitively inhibited. Gosh, that is the thing we would expect from low levels of a value, but certainly cannot be competitively or must be competitively are very strong extreme statements for something we know is variable. Immediately, we would have probably predicted answer choice B of the remaining ones left. And if we look at the actual diagram here, we have that sildenafil citrate is affecting that PDE5, which apparently is forcing it into the cGMP pathway. And we know, just going back for a second, that cGMP is this necessary first part that is leading to erection. And it must be the case that Viagra does not produce its own cGMP, otherwise it would just do that earlier in the process. Clearly Viagra does not replace it because cGMP still had to be produced in the first part of the process, which is all of that stuff about absence of sexual stimulation. If we didn't have that first two red arrows, then we wouldn't be getting to a point where cGMP must be made in order to finish out the process, in order to have that erection at the end. Notice, though, that last conversation that I just had with you about following the entire pathway, which way of tackling the problem seemed easier? Getting rid of patently false answers, or walking through that pathway to get to B? Which way seemed more attackable? Yeah. And this is often the case. That's why I bring it up, Quinley. Usually on a long, complicated pathway problem, getting rid of answer choices we know must be false can get you down to the one right answer or down to the 50-50 split with a lot less work than actually going through the entire pathway and looking for all of the questions that could potentially come up. Now, as far as this pathway is concerned, we can see a lot of questions on a complicated pathway. We might see questions that deal with enzyme kinetics. We might see a question about vasodilation, a question about membrane transport. All of those are common themes on the MCAT we might see based off of the general overall pathway we see described here on screen. For example, that membrane pathway or kinetics or a question about transport mechanisms, all of those would be basically discrete content areas that they could tie into the passage to make you go back and reference it. So it could still be a pseudo-discrete or purely require you to go back and look at the passage. But as a follow-up, I want to remind you, if we do have a complicated pathway like this, or we do have a very large diagram or flow chart, don't spend a lot of time trying to actively memorize it or get every detail. It wouldn't be within your time limits. It wouldn't be an efficient use of your time. Instead, get a gist for the overall figure, look at a couple details when the question asks about it, but otherwise, big picture, big flow diagram, jump into the questions. Let's do another question related back to NO producing stuff, because we just had that in the last question, and see if we can get the answer quicker than having to go back and look at the passage and the pathway. So help me out. Is a long, in-depth analysis of this figure necessary to answer the question? Do we have to go back to a passage information about other axons or neurons? No, Christopher says. We can go through and find the answer faster. I agree. Absolutely. Now, it's mentioning that NO, we'd have to know something about NO being released. Nitric oxide does something special. There certainly are neurotransmitters that are in the solid state, and the neurotransmitters in the solid state can act on neurons across a synaptic junction. But NO, is it a neurotransmitter? Better question. Is it a solid neurotransmitter? You might know this from going to the dentist. It's a gas. Oh, wait a second. Gas? Well, what can gases do if they're small? And what can gases do in general when they're released outside of a cell? They can diffuse. Interesting. So nitric oxide diffuses from the neurons. It's not passed via dendrites. So then, if we have this unlabeled neuron releasing nitric oxide, where could it go to if it's diffusing through tissue? Anyone that it wants to. Absolutely right, Christopher. Answer choice B, 1, 2, and 3. Now, I've got a bonus question. What if it had been acetylcholine? What if it had been a neurotransmitter that was a solid state? Now what? If we got this unlabeled one here, I'll put an X on it. What if that one had been releasing instead? Now where could it go to? It looks like it's to 2 and 3. Exactly. Quinn Lee, Christopher, because clearly, number 1, doesn't actually flow from the axon edge or the axon point, or again, our axon terminal, synaptic point, anything that you want to describe it as. It doesn't flow out of our X-labeled neuron here. Nicely done. Let's do a couple more just to get a chance to really lock in some of that conceptual knowledge from this nitric oxide one. If it's affecting the arteries around the circulatory system, what would happen? Well, first of all, this is a question where we don't really need the pathway. It's sort of a pseudo-discrete. Nitric oxide affects the arteries. Remind me, does that vasodilate or does that vasoconstrict? If you've got a lot of excess nitric oxide. Causes smooth muscles to relax. This is dilation. Absolutely, Christopher, Afshan, nicely done, Andrew. So if they're relaxing, dilating, then we're giving them more space to move around. They're going to be happier. It's going to cool off the body because we have more flow to the top surface of the skin. It's going to decrease venous pressure in the veins. It's going to decrease overall blood pressure. All of these would be classic answers. So how come I don't just jump in and circle A then? Vasodilation, cooling of the body. How come I don't jump in and circle C? Watch out. There's one word there in the question stem that if we had read too fast, bummer. It is an accept question, so the only one that should not be the case is an increase in blood pressure. We don't have any sort of squeezing or any constriction that would possibly increase blood pressure. Nicely done. Let's do 2 more. here we go. For number 9, follow up on that last one. Why might we have diffusing across a cell? Don't even look at the answers. What are 1 or 2 reasons we could easily have diffusion? This is the sort of assess, plan step that we would always do on any problem. Even without looking at the answers, because there might be some ones that look tricky. Small would be good, absolutely, Christopher, or nonpolar would be good. Take those in your execute step to the answer choices. A mentions polar, no. C mentions small. Oh, we didn't need the passage information. It's a pseudo-discrete. Passive diffusion, aka simple diffusion, is small or nonpolar. Boom. Answer choice B. Free radical, I hope not. That's super active and concentrated inside the cell isn't required, but also we wouldn't want to have a whole lot of nitric oxide concentrated in our cells, hopefully, because it could do some bad stuff if it all of a sudden spontaneously got sent out. All right, last one that I want to make sure that we cover in today's session. Let's take a look at number 10, a follow-up relationship. If Viagra is inhibiting phosphodiesterase 5, oh, that's that PDE5 thing. Okay, we might have to go back to the passage just to double check. But PDE5, well, okay, it's inhibiting something. Gosh, maybe I'm short on time. Maybe it's our last minute. Could we at least get rid of a couple answer choices that would not be an answer we'd expect from something inhibiting something else? I'm asking this in kind of a vague way, but I want you to be kind of critically thinking. What could we get rid of? If Viagra is inhibiting PDE5, what would we think we could probably get rid of that wouldn't make sense? What I'm asking, just to be clear, is are there any answer choices that patently should never be right? And I like that Peter and Sylvian both put in here B, because reducing the Vmax of the enzyme, the enzyme's Vmax is the value associated with that enzyme. The enzyme will always have a Vmax value even if it can't get to its Vmax. So that's patently false. We could not get answer choice B to be true. And I like what Christopher put in here as well, binding irreversibly to the active site. So Viagra is always going to be on those enzymes forever, irreversibly from here on out. So anybody who ever took Viagra would have that completely shutting down that enzyme for life. Sounds pretty extreme. Generally on biology questions, we don't usually have always something. Get rid of A. Now, C could be tempting. We might bind to an allosteric site and shut something off. That could inhibit, potentially. And then increasing Km of the enzyme, well, that could also potentially be true, right? Because if we're inhibiting this PDE thing, it could be influencing the Km. It's not going to change Vmax, but it could change Km. So in this case, we don't really need a big relationship back to the pathway. We don't really have to go back and look at it in depth, but it did mention in the passage, if we went back and just scanned quickly for that PDE5, that it competitively inhibits PDE5. So is competitive inhibition the allosteric site? Oh, bummer. It was tempting, but competitive inhibition had better be the active site. So it must be this Km thing, even if we weren't familiar with it, even if we didn't remember, just for our benefit. Remember, Km is the concentration of substrate that permits the enzyme to achieve half of its maximal velocity. So it's sort of a proxy measure for the affinity of the enzyme to the substrate. Lower the Km, the higher the affinity for the substrate. Lower Km, higher affinity. So increasing the Km of the enzyme makes sense, because if we've got competitive inhibitors, they're not going to affect Vmax. They're going to affect potentially the Km. Nicely done. If anybody has any follow-up questions about this, absolutely let us know in the QA chat. But we are perfectly on time here for tonight's session. Thank you for the Biochem review that we just did. And if there's anyone else who has follow-up questions, let us know. But I am going to be doing the CARS full length 2 immediately after this right now.